Okay, hi everyone, we'll get started. I'm Saket, and this course is an introduction to computational multiomics. Given this is lecture one, we wanted to sort of just set the ground straight in terms of what this course is going to be about. If you have any questions, what should you expect from this course, so that there are no surprises, right? So while life is very different, right? Like it will give you a lot of surprises in the end. But one motto that I have generally for courses is there should be no surprises. Um, so we lay down some of the expectations and what this course is really about. But this course, this is the third run of this course, which I'll introduce in just a bit. But one of the expectations is we'll keep this interactive, and it's a cliche when it comes to a lot of these classes. Everyone wants to keep it interactive, but I mean it in its truest sense, because some of the discussion that we have here gives me a ground to actually go back and think I will not know answers to a lot of your questions, but often a lot of new things and interesting things come out of those discussions. So I'll just want to keep it interactive. Okay, so the first thing that I wanted to start off with was this quote from Sidney Brenner. People know Sidney Brenner? Anyone know Sidney Brenner? A lot of people are not from the background of biology. Anyone knows who's Sidney Brenner? So Sidney Brenner won a Nobel Prize in 2002 for in physiology or medicine for mastering the use of C. elegans. It's an organism for biomedical use cases. Particularly, it's used in biology as a model organism. He says the big lesson to learn here is that in science, only mathematics is the art of perfect, physics is the art of optimal, and biology is the art of the satisfactory. If it works, you keep it. If it doesn't, you get rid of it. Okay? And you'll see once we start to do the analysis part of things, you'll realize what this quote actually means. So mathematics gives you this art of being exact. Physics gives you engines to optimize things, and biology is a science of observation and then drawing inference from it. We sit in an engineering school, which keeps us at a very special place in trying to understand biology, in the sense that while in your usual classes you think about attacking problems, the way that this course is designed is to actually help you to think. If you see something, why does it exist? What is the mechanism? Why is it the way it is? Right? We're not trying to sort of develop something which is a solution to solving a problem. We have to define why that problem exists to begin with. And that's the approach we are going to take. So I'll just talk about a brief outline of the course. Anyone recognizes this building? You are not allowed to answer, but it's chemical engineering, right? So I studied chemical engineering, so I'm also coming from, I'm not a domain expert, with a fair disclaimer. I had the idea of having a course like this when I actually went for grad school after doing chemical engineering here. And I took grad school at USC, where they had a very strong, very mathematically oriented course structure of teaching biology. So I joined in March 2024, and I always wanted to float that course, which I found I would have had a lot of fun learning from if I was an undergrad here. So all this course is designed with my personal flavor, and of course, I'm biased and I will make mistakes and so on, but hopefully you'll see that why this course exists in the format that it does. So this course was approved in pretty quickly, actually, in just a matter of 2 to 3 weeks. I floated it the second day when I joined because I wanted something to teach which I could relate to. And so in some sense, this is my baby, and this will always remain my favorite baby because it's my first baby and I don't have a baby. Right? So the approval came pretty quickly. We have had past runs of the course. Yeah. The first run, like any course, had a lot of people taking it. There were 57 people. It dropped down to 45, and I think the number is going to go down further. Some of this is, of course, you'll see that why do I deliberately do it. this course actually is not for everyone. I'm not trying to do gatekeeping. I think you've already received those emails. The idea is to have people who actually truly enjoy problem solving in its truest sense. And what do I mean by problem solving is, given a problem from a particular domain, are you able to critically think and attack this, irrespective of the background that you're coming from. So that's why this is the first year where I'm actually going to do some gatekeeping, and you'll realize what that means in half an hour from now, what that gatekeeping involves. But that's the entire motivation of having the course in its current form. This is the third time, so there'll be some new changes, some new things, and some old things that I've learned in the last 2 runs of the course. We'll get rid of it. Okay, so I think most of you probably would, any questions? Most of you probably would have this question about, this is the first question that I often get. How will the grading go about? So till last year, we used to have an assignment heavy course. We're going to ramp it down a bit just because, there have been a lot of changes in the world outside. And among other things, we have burnt our fingers doing assignments because you can practically, I can give you a solution and I can give you a problem and you'll have a solution in 5 minutes if you wanted to solve it. While that is of course welcome, and I'll talk about it because you can use any large language model out there to do this, the idea is not to emphasize that too much. So we have reduced the contribution of assignment and focused more on exams. We have n-1 out of n. This will all be due within 1 week after we release it. We'll use something called GradeScope. If you have not used it, you will receive emails once I confirm your registration. And it's fairly straightforward thing to use. Everything will be online, no paper submissions. You will never ever submit any paper physically except for mid-sems and end-sems. You will never get back a paper. It will everything will be online, including what's called the crib session and all those things. There's a late submission policy. You can submit up to 3 days late. There'll be a 10% penalty per day. So if it's due for 100 points or 100 marks, if I submitted 1 day late, 90%, you can only have a maximum score of 90, 80, and 70, and then we stop there. Okay. I'll talk about the LLM policy once I cover the next slide. You are allowed to discuss. Again, we are here to learn, and the best fun I have is to learn from you. And I'm sure you have it the same way, which is you learn from your peers the best. So you can discuss, just attribute if you discuss it with somebody. And when every year I mention this, I actually mean it in its truest sense that, when you go out of the world, when you do projects, when you write papers, you will of course have a co-author, right? You will have somebody whom you would have discussed the paper with, whose ideas might have come from somebody. It's perfectly fine, you should go ahead and acknowledge it. People often hold back, and then, we have to unfortunately do the policing thing, which is the most, it's the most unpleasant thing of part of the course where we have to do policing. We are not here for that, right? We are all here to learn. So hopefully we don't have to go through that policing route. Given that this is explicitly bulleted in the slide is for a reason. If you discuss it with somebody, just feel free to attribute it. Now, there's one caveat here that when you discuss somebody, your two solutions can still not be the same. It can never be the same. We don't work like that. There is a stochasticity involved. You can discuss, you come back, and you do your own solutions. It has to be your own work. Often people try to rationalize this that, I discussed it with this XYZ person and I submitted a solution. It's going to be the same code, right? No, it cannot be the same code. It doesn't work like that. Human mind doesn't work like that. Never ever can two people be thinking exactly in the same series of fashion. Like that just is unheard of. Okay? So don't, hopefully, we'll not have to do that policing thing here. I have very, very high hopes from this class. So let's keep it that way. Midsem is 25%. It's closed book. There's no collaboration. This will be offline. So is the endsem, which is also 25%. Again, closed book and offline. And the remaining part of it is course project. So the course project will involve overall contribution from, like, it's a group project. So all 3 members will have to do a course project which is a good avenue for you to actually put all the things that you learn in this class into an actual project which will come from you. So that's something which you will be able to do. This will start to talk about topics once you have discovered some parts of the course in late August. There'll be surprise quizzes of 10%, right? So usually, so in the last 2 years, I've not mandated attendance because I believe this is an elective. Nobody is forcing you to take this course. You're here because you're interested. As I wrote in the email, I cannot guarantee any grade, right? I cannot guarantee, you'll get XYZ grade. We are all here to learn, and I teach it from that perspective that you should have fun irrespective, and I'll take care of the rest, right? And when I say this, I mean it in its truest fashion, which means that if you're having fun, if you're doing things, you will do good, right? And I'll take care of that part. Okay, so to do that, I have to have some rules. So in a sense, it's a 100% attendance class. Every class that you miss, without, of course, a legible reason, which could, of course, include medical reasons, you'll incur a penalty of 3% from the total score. So if the final score is of 100, I miss 2 classes, I can only score a maximum of 94 points. There is one caveat, which is that if I miss 3 or more missed classes, I will have to assign a DX grade. I usually don't do this, and I never have done this, but just because of the changing times and everything that's going on, I think, and given that everybody here should be interested in the class, I have to enforce the policy of DX. Okay, so people who are new, DX essentially is a grade that gets assigned close to the deadline before your end sems, slightly well before end sems, where it is written that it won't go in your transcript, but essentially you will not be able to take the exam. You have to essentially drop it. Any questions about the policy? Okay. Final grades will be relative grading. It's called RG in other words, which means that even if the maximum score of the class is 70, doesn't mean that that person will get will get a BC, which is the average grade usually. But if it is maximum, of course, it should get the highest possible grade, and everybody else will be graded on the curve. Grading essentially on the curve means that if there is a distribution like this, people who are on the extremes will get some grade, and it will taper off. And that's usually an objective choice. Again, just to emphasize, I am on your side when it comes to this. Unfortunately, we have to follow some mechanisms to evaluate in some sense, and that's why we are doing it. And I guess one way to control this is to have a curve which looks flat, and I usually tend to do that, but there are no guarantees. Okay, any questions about this? Okay, so if you're auditing, some of you are auditing the course or might be auditing the course, the requirement on the last slide holds up, except that you will have to score everything with a passing grade, which is 35% of the maximum score. So each assignment, if the person who scores maximum is like 60, you have to have at least 35% of 60, which I think is fair. That's the passing. So you should complete your assignments with passing course. There's no course project, and you should either write an end sem or a mid sem with passing scores again. Okay. Any questions about this? Okay. So the course is a bit of roller coaster, right? There will be a lot of theory on the board, sometimes a lot of theory on the slides. There will be hands-on sessions, which are often very, very chaotic. But the course is designed to be like that because the real life, when you go out and actually handle data of this scale, it is going to be a roller coaster. I still make mistakes in my analysis, things still get a bit chaotic, but all of it is actually part of the course, right? So you have to be able to handle that level of roller coaster ride. So one of the things that will often happen is you might not remember what number of the course that you took. You might not remember this course. You might not remember my name. You might not remember most things that are taken part that you take part of in this course about. The only thing that you'll remember are things that you will do from scratch, which, for example, includes your assignments, exams, right, and your course project. So that's why there is this emphasis on a mix of assignments, course project, and exams, so that you remember and you relate to the content that is being taught in the class. The mid-sim is slightly going to be more on the theory side. The end-sim is a full-on open-ended exam, which only and only tests for your critical thinking. Can you actually take problems from biology, given problems from biology, can you actually think what would be going wrong? How would you test a particular mechanism? How would you test whether the system is working in the way that it is supposed to work, and so on. No theory questions usually in the last part of the exam. The second part of the exam, the mid-sem part of the exam does have theory. And all of it is for this reason that you will require some theory to actually do well in this subject overall. Right? I have a choice to not to glance over. Usually this course in a lot of institutes is often confused as a bioinformatics course, where you get to learn different techniques and different tools, and you run some of it and you think you understand. But often in the long run, those don't support you if you want to do deeper stuff with that kind of in that kind of field. So in any way, the kind of world where we live in, writing code is no longer, it's no longer useful. Well, it's a useful skill, but it's no longer something that will make you stand out. What will make you stand out is the ability to be able to critically think, to critically argue, and that's why the course focuses on those aspects. There's huge potential here to make an impact, which is you can have output a preprint, which means you can have a paper out of your project. You can have your open source bioinformatics package or anything else. And I'll just pause here and give you two examples of the approximately 90 plus people who have taken this course. I have two success stories to talk about. The first success story is a personal success in some sense, because I learned a lot in the process. This was a student who took this course in the first run, Dhananjay Raman. He's a computer science student. And during his project, he came up with an idea which I did not approve of initially, because I thought it's a very straightforward sort of idea which will somehow work, but it won't really add meaning. But he still went ahead with it, implemented it, showed that it is the most efficient solution out there. It still exists on our lab repository right now. We haven't actually published it, but for all practical reasons, it's a publishable solution. The reason it's a personal success in some sense is because he taught me a technique which afterwards I've used in a multiple different tools that we have developed in the lab. The same exact idea which he conceived of is something which I've been able to generalize across different solutions. So while I'm not sure if he relates to this idea or not, but personally I ended up learning a lot just because of that one project. The second project is, the second idea was success stories of another student who took it last semester, Ananya Chavdal. She was an aerospace student, had absolutely no relation to biology. Ended up with her course team. I ended up doing a project, worked with me separately on a totally different problem, but related problem from biology or health sciences in general. And ended up applying to grad school. Wasn't very sure, but got into one of the best programs out there, which is a mix of computer science, even though she's going to be in the computer science department. So she switched from aerospace to computer science, but her focus will be biomedical informatics. So those two stories are, it's small. It's a small number, but when I moved back, that's one thing which I wanted to be able to do, which is, even if there is one change that we can bring to the society, and I hope you will probably think the same way, we should all do it. And I hope this course will be useful to you in one way or the other. The way to define success is very personal. You define what is success for you. For me, those two people are something which define success in one fashion or the other. Okay. My slides are hung up. We'll talk about the project ideas in when the time comes right, but I'll just give you a broad flavor of what the projects usually involve. There's a bit of benchmarking and development. There is reanalysis of published data sets. There's application of statistical methods for solving problems in genomics. When I call something statistical methods, in quotes AI ML, right? Because that's what the modern version of it is for. Data integration, which will become clear once we talk about it, and your theme. You can bring your own project. While I will float a set of broad topics, you're welcome to work on a problem which you relate to, right? Again, when you bring your own problem, it will be relatable. You will, of course, by definition, you will end up doing good because it's a problem that you define. Okay, so the course idea, which I already mentioned, is to help you become a synthesizer and a critical thinker. This quote is from Edward O. Wilson. He worked a lot on ants, wrote a bunch of wonderful books. One such book influenced me a lot and influenced my decision to pursue grad school. He says, we are drowning in information. He wrote this around actually more than 50 years back. We are drowning in information while starving for wisdom. The world henceforth will be run by synthesizers, people able to put together the right information at the right time, think critically about it, and make important choices wisely. I think stands very well true in the modern times. You have a lot of tools that you have access to. So the USP of your knowledge will come from whether you are able to synthesize the information, right? You have access to all the information at fingertips. The time he wrote this book, I think the internet also did not even exist, right? So all you had access to was just books. And then he's talking about this information that there will be a lot of role that the synthesizers would play, which is something which I think holds true still in this modern world where you have access to all information at fingertips. So if you want to be relevant, and this holds true for people like me as well. For the research lab that I run works just using, one way to define it is we write code for biology. But if you think about it, part of our job is already done. There is Claude, there is ChatGPT, there is other LLMs. But one thing that still hasn't really arrived is to be able to think critically, to be able to make these broad connections. While LLMs are getting very, very close, but the experiential learning that a lot of humans have, it has a lot of meaning still, which is relevant in the modern world. Whether it will be true in the next 2 years, that I don't know. But as of now, and I use these tools very profusely, so I know I'm speaking from experience and not from a skeptical lens, that we still haven't arrived there. But maybe, LLMs will arrive. We are still relevant as humans. Okay, so we do have LLM policy, which is you're allowed to use large language models. You can insert any new models there. You should document when you submit your assignment what and why you're using the model for. Which questions did you use, and what was your original source in terms of, it came from ChatGPT, it came from Claude, and so on. What we expect, however, is you understand the solution that is being spit out by the LLM, right? So the LLM any assignment that I gave, that I will give, I'm sure it will be able to get like 80–90% on it. If it's able to solve Olympiad problems, it should be able to get very, very well. But what I expect you to do in the process is to understand the solutions that you're providing. While if the assignment is due at 12 midnight, you can submit at 11:55. Even if you ask Claude to start doing it at 11:50, it will be able to do it. But I don't think you have learned anything in the process, right? And if you want to do this, then unfortunately this course is not for you. The reason for that is you should be prepared to explain what you did in your work, right? So at any point of the time, and I'm going to use this slide as a leverage to say that at any point I can invoke my right to question you. If I see an assignment and we have some ideas about where the solution is coming from, often we will make mistakes, but most often we get it right. We have the right to invoke that we are going to ask you to come and explain how you arrived at a particular solution, or why did you approach it in that particular way. That said, I'll show you some examples where LLMs can actually make mistakes. So if you have those in your assignments and you don't declare it, it's straightforward. It's very clear that you don't have idea about what you're talking about, which is not a good situation for any one of us to be in. So try to avoid that. If any one of us detects the LLMs without attribution, you will not be graded and you'll be given a 0 directly. If you don't attribute and it's very clear that it is taken from an LLM, we'll directly give you straight 0. No questions, no discussion. Okay, for coding questions in the assignments, there's no partial marking because, unfortunately that era is gone. For theory questions, you can expect to, you should still be able to explain, you're still expected to explain your answer and your reasoning. Okay, we can randomly select a few students to come and explain their answer. This can happen during the class, after the class. Anyway, you're expected to be in the class all the time. So just be ready for that. This can also happen during your end sem, for example, or your mid sem. If we detect anything which we think is something that that doesn't seem that to be coming from you, we have the right to question it. Okay, is that fair? Okay. Okay, so the class, of course, is on Mondays, Tuesdays, and Thursdays, 5:05 to 6 p.m. Except the exception today, we'll have a 1.5 hour class because we missed the class on Monday. If you want snacks, tea, coffee, anything, you get it at 6:30, and I'll reimburse, right? Because you will not be able to go to the hostel. I think it ends at 6 p.m., right? Your tiffin, so you'll not be able to eat it. But please let me know. I'm happy to reimburse whatever it is. We will use, so my office hours will be on Wednesdays 4 to 5 because that slot usually is for where you don't have anything else going on. You can also use this link for appointments or email me. It's better just, if it's an office hour question, just better to just put the appointment on that link already. We'll not use Moodle. We will use something called Piazza for discussions. Once we approve your registrations, you will get a link to join it. The advantage of Piazza is it allows you to ask anonymous questions, which in the past has been very, very powerful because there is no fear of judgment or a pushback from any one side. Okay? But use it wisely and responsibly. All the course material is on this website. If you have a personal question, you can directly post to Piazza as well. It can be private between you and me. If you still don't feel comfortable, you're welcome to email me. If it's a question which you think will be generally asked, just ask directly on the forum, right? There'll be mistakes in my slides, or there'll be mistakes somewhere else. There'll be a clarification question of the assignment. You can open up open question on Piazza, which everyone can see, but you can still hide your identity, for example. Right? So you can do all of that. Okay, we have 3 TAs, Khyati, Sharda, and Ravi. They will all sit in Crescent building. Crescent is a building just literally opposite to the lecture hall complex. We sit in the basement. B20 is the number. There are only 2 rooms there. One is B20 and the other one is B22, where my office is. We just have, it's very, very close by. They all have their office hours on Mondays from 4 to 5. And go to their office hours if you have questions about assignments, anything going on in the class which you don't feel comfortable asking me, you're welcome to go and ask them directly. All of them have taken this course, so they will know what they're talking about. Okay. So there is no textbook for this class, but there are plenty of papers, and we will link them in the syllabus, which if you go on the website right now, you will see that it's a blank syllabus. But as we cover different teaching material, we should be able to see the different papers which are linked to a particular topic. The assignments are meant to, we will discuss some theory in the class, and we will have hands-on usually on Thursdays, where I will ask you to bring your laptops. We will do some of the exercises in the class. The reason to do this is to ensure that you actually implement some of the ideas that we discuss in the class through your code, right? Or take a data set and analyze it. The slides will be text. I will do some blackboard work once that time comes. There will be sufficient references, and it will become clear as I talk about the other slides. You can just click on that reference, and it will take you to the original source, which will allow you to read more if you have to. Okay, so this is not a coding class. We don't teach programming as such, but there are some material available. We will use a mix of Python and R depending on what is the requirement. If you click on this link, and you'll get the link to the slides on the course website. There are some programming resources. They are not comprehensive, but they will set you up for installing things that you will require for some of the sessions in the future. Most of this will gather speed sometime in 2nd or 3rd week of August. Okay? There'll be some theory which we need to do before you actually start to do the practical side of things. Okay, so I don't have to read out this slide. You're expected to work on your own, and you know all of these points. One simple thing that you should remember, the summary of this is just do the right thing. Internally, we all know what is the right thing, right? Your mind tells you, your heart will tell you what is the right thing or not. Don't let your brain try to rationalize the wrong thing. What is the right thing? Your heart will tell you. Don't let your brain win over. Just do the right thing, which means that, you know, don't cheat in exams, don't do anything which we have explicitly talked about. You have also, when you joined IIT Bombay, you also signed the policy, which I will just read out for clarification. I declare that I will adhere to all principles of academic honesty and integrity throughout my stay in the institute. I will not seek or give unauthorized assistance in tests, quizzes, examinations, or assignments. I will not misrepresent, fabricate, or falsify any idea, data, fact, source in my project submissions. I understand that any violation of the above will be cause for disciplinary action as per the rules and regulations of the institute. None of us want to be caught up there. Okay. Okay, right. So just to highlight, you know, don't try to game the system. We are all here to learn. You know, I don't want to be the police. None of the TAs, you know, fancy being police. We are all here to learn. So let's just do that. Okay. This is just again your the same policy repeated. So we'll start class on time, except few exceptions. If I have a meeting just before, I might run 5 minutes late, but I don't think I will run ever more than 5 minutes late. In the last 2 runs, I think we have started this class particularly on time always, right? It starts at 5:05. If you're asking a question on Piazza, be respectful, you know, maintain a collegial atmosphere, you know, don't try to, you know, we are all here to learn, right? So we are all on the same page, including me. Also, my name is, you know, my name is Saket. It's not professor, it's not sir, it's just Saket. Okay? It's simply, and this is my choice. I want and I prefer you if you call me by my name. Okay? Even if this is unusual to you, you know, if you say this to me, that is respect. Okay? So that is respect. So show your respect if you have to by taking my name, and don't prefix with anything or suffix it with anything else. Any questions? Okay. Now let's get started. So what is this course about? Biology in hard and slightly higher text font, or lengthier text font, because that will form the essence of the problems that we're going to talk about. So the problems will literally come from biology, and the solutions will come from which will involve mathematics, probability and statistics, and computer science, right? And you can see right here itself there is some bunch of stuff going on, because it is going to be a roller coaster. We'll pick some topic from here, we'll pick some ideas from here, but then the ultimate aim is to be able to solve problems in biology, right? So each subtopic that you're seeing here can in principle be a course in itself. There are courses in IIT Bombay which teach you linear algebra, there is a course on discrete mathematics, on calculus, probability theory, and so on. Of course, I can never do justice to any of these topics. What we will try to do justice to is to biology, by understanding the problem in a sense from first principles and trying to study it at scale. Computational biology as a subject is hard because you need to understand biology, but you need to also understand the other topics, right? And that's why it's a niche subject in itself, right? So in the scheme of studying these hard things, we will be able to realize, right? We might often lose the context of why did we even start. So sometimes when you teach such variety of topics, we will lose the essence of why are we doing this. I will try to keep emphasizing, and that's why you'll see I often repeat myself. That is to emphasize, and in this case, the emphasis will be on the original problem that we started with. So this is broadly called the reverse Bollywood approach, where you reveal, so Bollywood movies are boring in the sense that, you know, they start with a lot of build-up that happens before they deliver. We will do the other way around. We will deliver first. We will have the climax come out first by the actual problem that exists in biology, and then we will try to solve it using the tools that we have developed from mathematics or statistics or computer science. Okay, so the course is to give you also a flavor of science, and this is a deliberate choice, despite being in an engineering school, is to teach you how to do science. And this is not science that you must have done, all of you must have done in your class 9th, 10th, 11th, 12th. The idea is to be able to help you think the distinction between science and engineering, right? So the focus of science is to inquire, which is to have descriptions or mechanisms or explanations of why we observe what we observe, right? We are all alive. I can move my hand. How am I able to do it? That is a question that a scientist would ask. An engineer would try to sort of ask a different questions, that can I make something so that my hands move faster? That's a solution that you bring to it. You might not necessarily have to understand the mechanism of how my hand is moving, what neurons are firing in my brain to make my hand move faster, because you can bring a servo motor or you can bring a faster motor and just attach it, and I should be able to make my hands move faster. So that problem of being able to inquire, why does it exist, right? And it's a theoretical question, it's an academic question, but it's a fun exercise to do, because once you figure out the mechanism, maybe I don't need the motor itself. All I will have to do is go back, if it's just one neuron firing every time, I just attach something here and then everything is in control. I can do whatever I want. I don't need an extra motor at all. So the goal is to be able to equip you with fundamental analytical framework to answer your own questions. I will define some of those questions because I have faced them in the course of 10 years of working in the field, but you should be able to do this once you have this framework mastered. This quote is taken from Atul Gawande. He used to be the medical surgeon head of the US. I'll just read this quote aloud. Science is not a major or a career. It is a commitment to a systematic way of thinking, an allegiance to a way of building knowledge and explaining the universe through testing and factual observation. Right? So hopefully everything is clear. You have a question, you have a abstract theory or model in mind. You first try to see there is a physical system that we observe these things in. We measure these things, we come up with a concrete description of the data, and then we try to compare it to come up with an explanation of why it is happening the way it is happening. That is science. In case of an engineering design, you already have an abstract model in your head. You try to distill the information, you try to describe it. I try to describe that I want to move my hand faster. You then somehow come up with a physical system, which is like a useful product, a motor attached to my hand, and you deliver a solution here. Here, in this case, I'm already observing my hand is moving. I'm going to ask a meta question, why is it happening? And then try to discover a theory that there are neurons in the brain which are firing in a particular way for you to be able to develop a solution to it. Right? Is there a distinction between science and engineering clear? Okay. So the course is about being able to decipher fundamental principles of how genes are regulated. We will study a different number of techniques where we go inside the cell and we try to measure what is happening inside the cell, right? So there is DNA, there is RNA, there is protein. If you want to study something, you want to be able to, a way to capture it. So there is some technology development that happens here, which is how do you profile the DNA in the cell? How do you profile the RNA in the cell? How do you profile the protein in the cell? You can do this. There are a lot of technologies out there, and I will strongly encourage that all of you also, at some day I'll hopefully see some of you developing in this space. Once you measure it, it is a complicated set of data because there'll be a lot of noise in biology. So you need methods. You need computational methods to decipher what is biology, what is technology, what is technological noise, what is biological variation, and so on. So we will develop a framework to identify these factors, right? The overall goal of biology in a very hidden fashion is to be able to identify factors which lead to human diseases. Why do I get cancer? Why do I have fatty liver? Why do I have diabetes? Why does somebody else doesn't have it? And even though they are eating glucose and sugar every day, right? Like why do we have so much disparity when it comes to human diseases? Why people from certain background are prone to certain kind of diseases? All of this in the essence is happening inside the cell. And the idea is to be able to decipher why is it happening at the molecular level. Okay? At the end of the course, one thing I will promise is you will be able to handle large and small scale data sets coming from biology in doing the analysis yourself, right? So a lot of people have later on come and told me where, especially people from biology background who could not run their data analysis themselves, that now they're able to do this and they can handle these different data sets. It might not necessarily apply to you, but that's something which I wanted to just mention. What is this course not about? We know what the course is about. We should also know what this is not about. It's not about using tools in bioinformatics. You can use it. You can use one tool versus the other, right? And we'll do some of it, but the course is not designed to teach you how to run XYZ tool on terminal or on command line. It's also not a course for answering questions in your bioinformatics questions, if you have done bioinformatics earlier, which go unanswered on forums. It is also not a course about how do I use XYZ tool to do my data analysis. It's a course about fundamental first principles approach to solving problems in biology. I will not help you be able to understand. I will not be able to answer questions about how should I run XYZ tool, because this is not a course about that, right? Or I'm getting a particular error in installing my favorite tool. How should I do it? I want to learn R, Python, or C++. This course is not for that. If you want to learn all cell biology or all of genomics or biochemistry, unfortunately, also this course is not for you, because we will touch on some principles. Nobody will be able to do full justice while teaching you both cell biology or genomics and first principles problem solving. Okay, so what is the role of computer science in or statistics in solving molecular biology? This is now a dated paper, came out in 2017, which says that all biology now is computational biology, because any measurement that is often now done, a lot of it makes no sense unless you can quantify it, or you can come up with a method or an approach to be able to understand, to help you understand why does the data look like that. Right? So you can go and read this paper, right? So this says that computational biology lets you see the big picture that exists in this domain. So why you might want to take the course, and this will be a good stopping point for a short quiz. It's a blend of mathematics, statistics, computer science, software engineering, and biology. You can apply first principles approach to solving biological problems. Genomics is something which is ubiquitous abroad. Those of you who are planning to go for grad schools or planning to work for companies outside, you can work in academic labs. You can go and work in industries, Genentech, 10x Genomics, Illumina. This is not an exhaustive list. All the pharma companies, for example, are missing. A lot of pharma companies do use a lot of genomics in designing drugs, for example. Right? It's also becoming ubiquitous in India. There are, of course, academic labs which use it, which develop it. There are a lot of actually industries in India also, including Strand, MedGenome, MapMyGenome, Precision Health, which are developing tools and products based on genomic principles overall. Okay, so we have a short quiz. It's an assignment. This is not graded, but this will be used for approving your registrations. I want to just get a sense of why are you here, what do you know about biology, and what are you excited about. Okay, I'm not trying to be the judge here. This is not a judgment quiz, but this is just to get a sense of understanding. Okay, we'll take 5 minutes initially. I think 5 minutes, that's how much it should take you. And there are 5 questions. You should be able to answer 1 question in every minute. Yeah. Also ask him to switch on the AC. It's very hot. if you feel like looking into your neighbors for this one, you should not be here. There's curious questions, okay? There's no judgment at all. Just be you and answer it. So the airplane question, it's the dots are where you see the bullets. These are planes which have come back from Europe. You are based in, or Germany to be specific. You are a statistician, and let's call you Abraham Wald. You have to tell that if you want to save more planes, where should you put the armor? Armor is something which is like the protective shield for protecting the airplane. And the dots is, each dot is an indication of where it was hit by a bullet. Higher density means higher number of bullets that would have hit. Yes. Only one armor. Only one armor. You have to be cheap, right? Or cost effective. Only one armor. Sorry. But it will cost you a lot. No, you can coat it with a lot of bullet. You can coat the entire stuff with bullet. But okay, think, right? If you do this, it's going to be like if you add armor everywhere, it's going to be very heavy. If it's an armor like a bulletproof jacket kind of thing, or they are heavier, right? You must have seen those are heavier. So if you add it everywhere, it's going to add. The airplane might not even fly. So you can only put it at one place. If you are constrained by that, where should you put it, and why? The rationale for that question is to think from first principles. So I saw a mechanism. Why is it like that? Should we stop? Need more time? Okay. Pen? No, you have pen? Hati, do you have extra pen? I'm also not carrying. You have okay, you can redo it and just write the correct estimate. Also, most answers just expect like 2 or 3 lines, not more than that, including the question on biology. You can just mention anything that you like about, if you. So the reason is I'm asking that question is because most of you would have taken BB 101 or an equivalent course, which is a introduction to molecular biology or introduction to cell biology and so on. Make sure you write your name and roll number. You can put a range. Range is fine. But the range should not be more than 10%. Otherwise, that has no meaning. So as I said, right, like the question, it's designed to just get a flavor about where the background that you are coming from. So you don't have to cheat. If you're cheating here, you're in trouble already. Don't do that. Okay, I guess it's 6, so we technically should also stop, but we'll have, I'll just take 25 more minutes. And for those of you who might find it confusing in terms of what is this course really about, I will cover what's called course vignettes, which is a description of like the broad problems that we'll be working with. And you'll see that all of these problems are problems from biology or health. And the solutions that are coming from are solutions from statistics, mathematics, or data structures, or algorithms, right? So they are varied solutions, but the problems originally come from biology. Okay. I think we should stop. Otherwise, there's no way we will be able to stop. Finish it. It's fine. You can drop it in the office. Okay. So, right. So what does the course really expect? It needs to have some exposure to biology, but it's great, but it's not really necessary, because biology is something which you can learn, even after. I'm learning biology still, right? So you can keep learning biology. Some prior exposure to mathematics is useful. It's possible that some of you have not taken math courses in the last 2, 3, 4, 5, 6 years, and that's fine. But then just be ready to put in more effort and time if the material is extremely new to you, right? That will happen. There'll be few topics where you might have to put in extra time. You should please ask questions, right? The best way to learn is keep your mind engaged, which is to ask a lot of questions and not make this a passive class. If you make this a passive class, I might make you sleep, right? So if you keep this engaged, you will learn more and I will learn even more. The other only expectation is not speak, write, use things which you do not understand, right? So if you don't understand, it's okay to say that I don't know, but even in your exams or in vivas or in your project presentation, if you don't know, it's okay to say I don't know, rather than trying to say that I know everything and then say incorrect things. Do not use brute force to make things work somehow. This is a useful exercise. I learned it the hard way when I started my grad school, because I was trained as an engineer. And I think I still have some essence of engineering left in me, which is to try all possible solutions and somehow solve a problem. More useful thing is to really pause, take a deep breath, and understand what is the problem asking, and then try to answer it. Okay? Any questions? Okay, it's not ended yet. But any questions for the content of the course, for the logistics of the course? The content I'm going to talk about in just a bit. Yeah, I could put in some but the usual advice there is OCW, which is the open, that is a good resource. We also have our own NPTEL actually. You should use that as well. Linear algebra courses there I think are good. We will use some of it. So you don't need like a strong deep dive into every topics in linear algebra, but if you understand what are matrices, what are some operations that happen on matrices, I think that should broadly suffice. Okay, any other questions? Okay, so what is the goal for today? In the next 25 minutes or 20 minutes, I will give you a 20,000 feet overview. What is 20,000 feet? When you fly in the airplane, you cannot see each and every building, but you can broadly see that there is a forest, there is a lake, and there are houses and so on. And then I will do, if there is time left, which I don't think we'll be able to cover, which is what is you're going to lecture to be about, which is the shortest introduction to molecular biology, which will help you bring all of us onto the same page, right? So we understand the language. But why should I care about all of this? Is we want to understand at the molecular level, how do cells operate? Once we understand how cells operate, we know how a tissue functions. If we understand something, how it works, we know when does it not work. So if I get a disease, for example, when do I get a disease? What happens? What goes wrong? Now, those are the objectives that we are trying to understand. One question that will keep recurring in this blog, in this course, and I was thinking about a blog because for other reasons, but the idea was this question about where did we come from is a very fundamental question, right? So where did I come from? Why was I, how was I born? Why was I born? Why was I born here? Where did my ancestors come from? It's a recurring question that will keep coming into this class. It will come questioning, where you came from, but also where your cells came from, where the diseases come from, and so on, right? So just remember this last question. Okay, we'll talk about what and how will we solve some biological problems in the course. The first problem is a problem of genetics. Before we do genetics, these are broadly but non-exhaustive list of topics which we will use to solve problems in biology. We'll do some probabilities and expectations. We will do principal component analysis. This is a standard technique that I teach in every class that I teach. We will do normalization. We will do some Markov models, simple and hidden. We'll do linear regression, generalized linear models, maximum likelihood, expectation maximization, data compression, spatial statistics, right? So you can see already there's a mouthful bunch of techniques. We only have few weeks to finish this, so this will never be comprehensive, but this will give you a touchdown on how tools from a different field, which is mathematics and statistics, is required to solve problems in biology. One such problem is of the faults in our DNA. The first one such fault is of having sickle cell anemia. So sickle cell anemia is a disease where the red blood cells, which should look like this round jelly-shaped cells, start to look more like this sickle-shaped cell. What happens essentially in these people, and you're essentially kind of born with it in some sense, and if this is not treated, or if you don't have a treatment right at the start, at some point these cells, because they cannot function the same way as your oxygenated cells, this will have trouble circulating in your body. At some point, this will start to swell up your arteries and your veins, and they might rupture, and it leads to, unfortunately, it can be lethal. What is very interesting about this process is it emerges from a single mutation, a single change in the DNA. So your DNA is made up of, we'll cover this in the class in detail. It's covered of these four bases: adenine, thymine, guanine, cytosine. Again, a bit of mouthful, but for practical reasons, it's made up of these four letters. So your DNA and my DNA, for example, what you might have answered in the class, how similar is my DNA and your DNA? Any idea? Yeah, it's very, very similar. 99.99, 99.9% similar. Right? So that 0.01% is what makes you you and what makes me me. While we share a lot of possibilities in terms of what can go wrong or what can happen, our sequence. So if this is the normal, let me call it ATCC. What can happen in the case of a particular disease is this T can get mutated to a C or a G or an A, and this is called a mutation. Every cell in your body when you're born has exactly the same DNA, right? Except you forget about the gametes, you forget about other cells, but if you forget on what's called your other body cells, right, like your skin, your hair, your liver, your kidney, your tongue, everything is the same, exact same DNA sequence, because you're bound out of one cell at a point of time. In such patients, however, what happens is this is your normal sequence, which is CTC, and we will learn this, that there is a complementary sequence to it, so that C complements with G and A complements with a T. In such patients, what happens is instead of having a CTC, you have a CAC. So the T has mutated into A, which leads to a different version of the protein of the hemoglobin. This is called the hemoglobin gene, HBA and HBB, alpha and beta, sorry. Because of this hybrid, because of this mutation, the protein that you're going to make is going to be slightly different. This is DNA, this is protein. Because of this protein, you get the function of the cell. In this case, the polymer or the cell that you get has, the protein that you get has a different shape, by proxy of which you end up getting a different cell altogether, different morphology of the cell altogether, which leads to a disease. A couple of years back, FDA, which is the federal authority on which approves drugs in the US, approved a therapy which fixes this, right? How will you fix this? You're all engineers or some version of engineers or scientists. How will you fix this problem? A person has a mutation at a particular base. What should have been A becomes a T. How do I solve this? It's an engineering problem. Yeah, find a way to switch the base, right? If I can somehow go there and I can switch this T to A to back to A, I have my original protein or original cell. That's not exactly what the solution that the approved therapy came up with was. Any guesses why? I should be able to go and fix this, right? Okay. Okay. Okay, good, very good, right? So you will have to go and make a permanent edit in this case, so that you and do it in your in your stem cells, for example, if you want to prevent this. But let's say we have a way to mechanism to deliver it only to the blood cells, and we can do it. Why is it still not possible to do it? It can have multiple side effects, right? So if I do this particular, so we are all assuming that we have that golden tool to go and make that edit, but actually that tool does not, it exists, but it is not yet at this scale. It is getting there, actually. It's getting there where I can choose a site where I go and I do a mutation of my choice, I can make it persistent if I want to, because it will be a permanent edit once you make it in the DNA. The solution was something different. We will talk about it once we talk about what made it possible, but broadly it became possible because of CRISPR-Cas9, a system which you probably would have heard of, earned the Nobel Prize a few years ago to Jennifer Doudna and Charpentier. The idea here is there is an enzyme which can shut down anything that you want by making an edit. We will discuss this. But only just this, knowing this would not have been possible if we did not know that what is the mutation, where does it happen. If I did not know to begin with that what is different between sickle cell anemia patients and the patients who are, or the people who are normal, that in itself is a problem of statistics. Like, how do you figure out what is different between two different sets of people, what is different in terms of their genetic sequences. Okay, there's another such disease which is called a KIT. There's a mutation that happens in the KIT gene. If you get a KIT gene mutation at a particular site, you get this white mark on your head. You can see the mouse has exactly the same mutation. And a lot of organisms that we use in the lab, for example, you will see a lot of people working on different variety of organisms. People work on yeast, mouse, rat. And the reason for that is because we do share a lot of sequence similarity with other organisms. How much do we share with banana? How much? It's very close, right? So it's 70 to 90% of it is very, very similar. How about chimpanzee? It depends on how you count, but broadly 98, 99. We're very, very similar in that sense, right? So how do you count it actually makes it slightly different, whether you're talking about the protein coding regions or you're talking about the non-coding regions, but that number ballpark around revolves around there. Okay. So the future of medicine, which often gets this buzzword called personalized medicine, is almost here. And Baby Muldoon is an example of that. Baby Muldoon was born with a genetic defect, which prevents the liver from synthesizing the nitrogen that often it is supposed to synthesize. In this case, what happened was the baby itself inherited a mutation which led to, in this particular gene, which is called the carbonyl phosphate synthetase, CPS1 gene for short. Because of this particular gene, ideally your body should break down the protein and produce nitrogen. In this case, once you get a mutation in this particular gene, that does not happen. If ammonia or if nitrogen primarily gets accumulated in the brain, in the liver or in the blood, at some point your brain will start to suffocate and it can be lethal. In his case, little KJ's case, it was a lucky discovery that happened as he was conceived. And in an ideal case, the way to solve this is you will swap out the liver, because the liver you're born with has this mutation which is preventing it from producing, or has a wrong mutation in CPS1, which is preventing you to produce or synthesize the nitrogen. But any liver transplant would take years to happen. And the solution that came from is they could actually sequence his genome or sequence his DNA. And in the past few years, we know how to make edits at a much precise manner. So you can actually go and develop a solution, develop what's called a personalized CRISPR treatment solution. All of this became, of course, possible because of CRISPR, but also the ability to decipher that where are these changes required to reverse the ill effects of a disease like this. There's also a story from India, which I will strongly encourage you to read. Unfortunately, while fortunately we could solve it in baby KJ's case, we could not solve it in Uditi's case. Uditi was born with a set of mutations. When I'm born with a set of mutations, often I don't show, there are certain set of mutations where you don't show symptoms in first few years of your life. At 9 years of life, she started to show symptoms of epilepsy because there was a single base change in a gene that codes for a protein, which is called neuroserpin or neuroserpin, which causes the polymers in her brain to become tangled. And because of this, you get epileptic seizures. Right? there was a lab in India which was planning to develop a solution for this, very much on the lines of what was done. This is 2012 or 13 that I'm talking about, actually 2018 that I'm talking about. However, by the time that solution could be developed, she unfortunately passed away. The reason to show this slide is to show how much of an impact you can make if you have a tool at hand and if you have mastered the art of actually learning what is going wrong in the biology. So my hope is at some point we will have our own solutions coming out from India for diseases, which includes what Uditi had, but also broadly the set of diseases that ails India as a whole. Okay, the way to solve some of this problem is to be able to align sequences. If you align two DNA sequences, you know what is similar and you know what is dissimilar. The first part of the course and the first few lectures will focus on how to do this alignment. What you're seeing, for example, here is alignment of proteins across different organisms. You can see that they are very, very similar. So whether I go to human or I go to lizard, this protein sequence broadly, which has the same color, means they are constrained, which they are same, exactly the same. How do we do this? We develop, take tools from computer science, including dynamic programming and some sequence statistics. So it's a mix of a CS and a math problem, that how do you come up with the most optimal solution to do this kind of alignment. Okay. You must have heard, or if you're coming from biology, you must have heard of this tool called BLAST. If you have taken BB 101, maybe it is covered, right? So often the way BLAST is taught is you go to the website, you input some sequences and you run it. This course is not about that. This course is about when you run that button, when you click that button, what is happening at the background, what algorithms are running. So it's a beautiful problem again, uses dynamic programming and again sequence statistics. Part of it is how any alignment tool generally works in the modern times. This is where we will start to get into the modern world. So BLAST is great, it works, everything is perfect, but we have moved on and now we are able to synthesize new sequences. This is done by this technology called sequence by synthesis, or the next generation sequencing. How you sequence this DNA to begin with. So if somebody has a disease, if I have cancer, if I want to know what mutations I have, the first thing that I should be able to do is sequence my genome. So we will spend some time learning the biochemistry, little bit of biochemistry, to understand how this is done in principle. And the reason to do this is, now when we started doing this around 26 years ago, it cost me $100 million to sequence one genome. That cost is now approximately down to $100 in 20 years, and it breaks what's called the Moore's law. People have studied Moore's law. What is Moore's law? Silicon, yeah, the number of transistors on your silicon chips on your laptop, for example, double every year. So it is this defies exponential laws of biology, and you will see a couple of more examples from biology which defies Moore's law. It's the baseline often people use for comparing growth. In this case, lot of technological advancements have made this possible. We will study some of those technological advancements. Okay, once we have been able to sequence the DNA, we will try to understand how do we align it to create to do this problem at scale. If I sequence the genome of all these people in this class, how do I actually draw inference from it in my laptop, right? These are like extremely long sequences. Our human genome is 1 billion base pairs. If I convert this to gigabytes, it's 3 GB, but when you sequence, you will get a lot more data. You will get data which is maybe 30 GB plus. Imagine doing this for all the class and being able to wanting to draw inferences from it on your laptop. How do you make this problem fit in memory and do inference at scale, which we're going to cover as part of this particular method. We will do something about transcriptomics, where we will start to study the RNA. So you had a question about drawing arrows which are feasible. So which arrows are feasible and which ones are not? Okay, so DNA gets transcribed into RNA, then Okay, RNA goes to protein. What other? RNA to RNA to DNA. Okay. When does this happen? RT. Yes, correct. RT. Okay. And off. Okay. And any arrows missing? RNA replication. Is there something called RNA replication? Can you replicate RNA? So biology is a science of exceptions, right? In principle, there are few places where this can happen. You can actually see a replication of an RNA. There are now recent papers which show that you can actually go from here to here, but the main thing is it's often once you land up here, this is your end point. There's no way to go back. There's no way to go back, usually, right? There are always ways that will, of course, in biology is like chemistry, I guess, is the science of exceptions. There will always be exceptions, but this is what is broadly called the central dogma of molecular biology, which we'll study in the next lecture. We will study how we can align data from RNA, how we can use data from RNA, and how do we actually study how genes are expressed. We will broadly try to study how to process this scale of data, and that's where the linear algebra part of it kicks in. How to analyze data coming from different groups, how to do exploratory data analysis. The reason this is the science of big data is how many genes do we have? How many genes in humans? How many genes code for proteins? How much? 25 to 30,000 protein coding genes. I'll just call it PC. So if I sequence this class, if I do transcriptomics, if I study the RNA of this class, I will have a matrix which is approximately 30,000 cross, let's say, if this class is of 50, it's a big matrix. How do I make sense of this large scale data? That's what the pre-processing part of it will cover. This includes techniques like linear dimensionality reduction, which I mentioned. It uses ideas of PCA, SVD, t-SNE, and UMAP. All of this will become once we talk about it. This comes from statistics. And then we do a lot of clustering, which also involves a lot of statistics. We will perform some testing, hypothesis testing. Again, a lot of ideas from statistics of how to analyze data from controls versus cases, or cases which includes diseases versus controls. If I want to compare these two people, how do I actually do this in a statistically robust fashion? Then we'll talk about single cell transcriptomics. This is a favorite topic because we work broadly in this in our research area. We try to understand what happens to each single cell. This is, for example, a UMAP representation of single cells in the brain taken from a mouse. Each dot here is one single cell. How do you draw inferences from, let's say, the mouse brain to the rat brain or to the human brain? So we'll do something called canonical correlation analysis and dimensionality reduction. Again, techniques taken from stats. We will see again a defying curve, which defies the Moore's law, which is how the scale of data themselves have grown, and the different number of statistical tools that have been developed to tackle this problem of large scale biology. So here the problem was 30,000 cross 500. We are now in the era of 30,000 cross 100 million cells. How do you handle a matrix of that scale on your laptops? That's something which we're going to study. Okay, we will study how to do causal inference in a very loose fashion. If I want to study the mechanism of one particular gene, the simplest experiment that I can do is I can do a knockoff. I can knock out that particular gene and then measure my expression again. Once you do this, that's the goal. That's a biological thing that you want to understand. But we have to develop technology. So we'll cover some of the technologies which are used for doing these knockouts. These are called perturbation experiments. And then we talk about what statistical models are used for doing causality analysis in these settings. We will perform some single cell multiomics. What you see here is the cell. It's made up of intracellular proteins. There is DNA, there is RNA. There are so many different techniques to measure it. How do we draw inference from the cell as a whole is the question that we are going to solve. And we will do this using along different modalities. We will study what's called broadly epigenetics. Epigenetics means something which is above the sequence. So when I draw the sequences, there are also some marks which are chemical marks that can get deposited. This is broadly studied using epigenetics. We're going to talk about some of these assays. And then the statistics of that. How is it different from just doing an alignment? What kind of statistical techniques do you need to study if I want to compare things which have this mark versus those which do not? Okay, we'll study aging and how do you define aging, right? So there's no one definition, unfortunately, that the entire field agrees upon. But because of a lot of money coming into the picture now, including with people like this who have sort of become biohackers and try to hack their own son's blood, trying to be young forever. We will try to study how much of it is snake oil and how much of it is actually real. Can you actually measure aging from a molecular perspective? What does it actually mean to be able to do that? Right? So people know this person. Of course, a lot of people would have, he made a lot of news when he came to India last year and left an interview midway because of, PM 2.5 and so on. There's good message that way, but I think that the kind of stuff that we are doing with our body without actually knowing the science behind it is actually not correct. So we should not do that. But that's my personal opinion. Okay, so we will look at some statistical models for doing this molecular level. This science is very, very new. It came out in 2012. Came out from a statistician's lab who were literally just looking at the data. How do you develop clocks which tells you how much is my epigenetic age versus how much of it is my real age? And it appears that you can actually measure this molecularly. I can tell, for example, that I am a 45-year-old caught in a body of 35-year-old because my molecular marks look like that. Okay, we will do this is new, by the way. We have not done this in the past. We will cover some statistical models for understanding spatial variation, that how does overall changes in the spatial characteristics. So if I have a tissue like my hand, are all the genes expressed the same way here or are they different? And there we'll do some spatial statistics. We will borrow a lot of ideas from spatial statistics to do this. We will perform and study some gene-wide association studies. These lectures will be guest lectures because I'm not an expert in this area. We will have a couple of people come and talk about how do you measure why Indians, for example, have higher predisposition to type 2 diabetes. How do you actually characterize that from a pool of population? We can also use the same data and similar methods to understand where did we come from. Are we genetically pure? Did India give rise to the rest of the world, or did we come from somewhere else, right? And there's always a lot of debate, and I don't unfortunately want to go, and fortunately I don't want to go into that debate. But there's a lot that the science tells us where we actually came from, which is much more believable rather than what the other groups in our country would like you to know, right? So we are actually genetic khichdi of the ancestors. We came from everywhere. It's not that, and partly from Iran's side. It's not that we did not give rise to the world. We are not the drivers of the world, unfortunately. Okay. Right, so I think I've already covered the spatial biology, but we'll go into the technology of this a bit. And then finally, and this is my last slide, we'll cover some deep learning applications that have found their way into biology, that once you have this scale of data, why traditional statistics often might not be sufficient. Okay, any questions? Any questions about the content, anything that was not clear? Okay, so I think I owe you a snack because I made you stay. So is this open? Canteen is open? No. The LHC one is not open yet. Okay, so we can go to Cafe 92 if you want to stick around and have tea and coffee or snacks, whatever you want to have. That's on me. If you have any questions about the class or course, please let me know. Your registrations will be approved conditioned on me going through your submissions. If it is approved, you will already know because you will get an email from Piazza. If it is not approved, I will email you, and we should have a discussion. Right? I'm not trying to gatekeep. I'm just trying to make sure that we are all on the same page and you benefit from staying in the class. Okay, thank you.